Hi, everybody. Thanks so much for coming. You're in the symposium on species interaction, kind of responding to human change. We're going to get started in just a little bit. There's the starting chime. The really big thing that we want to point out is after this, we're going to have a little happy hour at Bar Pamplemousse, which is about 15 minutes up the hill. So continue to talk basically about species changes in a rapidly changing world. And I'm going to pass it over first to Pryce, excuse me, who's coming to us from California State University, Northridge. Thank you, Pryce.
Hello, everybody. Thank you for coming. So I'm going to talk to you today about my master's research so far, which is on Joshua trees and their mutualisms with yucca moths. So I just want to start out today talking about just mutualisms in general. So I just have an example here of like an idealized mutualism. So mutualisms are when two species interact and they both gain a benefit from that interaction. So right here we have a butterfly and a flowering bush, and these little circles represent like where they can theoretically live. And if these are mutualistic species, where they overlap is where this mutualism can take place. If these are generalist species, they might have other interactions outside of this mutualism. But if they're obligate species, so they're dependent on each other, they would only exist where both species can coexist. But these mutualisms are at an increased threat from climate change because if they are obligate, they are dependent on each other. So if one goes extinct, the other one will also follow. So climate change might be disrupting where different species can live in different ways. So this might lead to disruptions and extinctions. So the plant groups I'm working with are yuccas, and they have this very interesting mutualism with yucca moths. So when these plants flower, yucca moths will visit and they will pollinate with special appendages that they have. And then while they're pollinating, they will lay eggs in the flowers. These eggs will, as the fruit are developing, the eggs will hatch and the larvae will crawl out and eat some of the seeds that the yuccas are growing. And then once the larvae are fully developed, they'll crawl out of the fruit, fall right to the ground, dig into the soil, and they'll cocoon themselves underground and wait until these plants flower again. So the species I'm working with specifically, there's two species, the western and eastern Joshua trees. They live out in California, Nevada, Utah, and Arizona. There's two species, but they inhabit very similar climate niches. So there's the western species, Yucca brevifolia, and they're pollinated by Tegeticula synthetica, and the eastern Joshua tree, Yucca jageriana, and they're pollinated by Tegeticula antithetica. So these trees are very unique, and they're iconic of the Mojave if you've ever been out there. They don't flower every year. There can be years between flowerings, and these moths will just sit underground and wait until these trees flower again. So climate change, we know, is going to affect these trees. Their ranges will be reduced depending on how severe climate change is. The modeling shows between a 20 and 90% reduction depending on how severe climate change gets. You'll see here is a map of Joshua Tree National Park with the current range of Joshua trees and where they exist, and then our moderate emission scenarios. This is how severely they'll be restricted at the southern part of their range. But what do we know about their moths, and how will their moths respond? Well, we don't really know much about these moths, actually, because they're very hard to track. So we know they enter a diapause state in early summer underground. They'll wait in the state for many, many years. We don't actually know the upper limit of that, how long they can wait. Tracking them is really hard. We only see them when the trees are flowering. They don't respond to light or scent traps like other moth species do. But trees only make fruit when they receive pollinator services, so it might be a good way to track these moths using fruiting. So this diapause that I mentioned, if you're not familiar with it, it's a state that many insects go into. It's like a suspended animation state. Insects can enter it at any stage of their life cycle. You might be familiar with monarch butterflies. They'll go fly down to Mexico in the winter and hibernate in trees, and then in the spring they'll wake up and emerge when they receive the right cues. And that's what these yucca moths are doing. So many different species use different cues to avoid unfavorable conditions. So many species use changes in lengths of day, changes in temperature, rainfall, or even vibrations of their conspecifics to know when to wake up. But many, many species use cold temperatures as an indicator for seasonal changes. So once they receive cold temperatures and then it warms back up, they'll emerge. So warm winters might disrupt this cycling. So we're trying to figure that out. So to start, if I'm going to try and track fruiting, we should figure out why do these trees flower. And a recent paper out of our lab from Yoda et al. In Press, should be coming out in Ecology Letters soon. They figured out what is actually cueing these trees to flower. So this is like one of the graphs that they have in the paper that I'll present. So the y-axis is the partial predictor effects of different weather cues that they used in their modeling, and then the x-axis is just the range of those effects. So what causes Joshua trees to flower? Flowering is more likely when a wetter year follows a drier year, when winter temperatures are above 0 degrees Celsius, when maximum vapor pressure deficit is relatively low, and that's just a proxy for drought stress, and that vapor pressure deficit and temperatures have been relatively stable in the previous year. So how I'm going to collect this data is we've been using iNaturalist. So if you don't know, it's just a community science app. You can take a picture of any species, and then it gets uploaded to the site. It records when you took it, where you took it, and it has a lot of valuable information for us. So if you look up here, there's a picture of this Joshua tree that someone took, and if you look at the very top, there is actually some fruit at the top of this tree. So this picture was taken in 2008, and so now we know that moths were actually present in this location in this year at this time. So we're using this data, so we went through all of iNaturalist's observations of Joshua trees that people have ever uploaded. As of June, there was about 14,000 observations, and we've categorized them whether they showed fruiting, flowering, or just no evidence of anything. And then we have a different flowering and fruiting ranges. The percentage of each and any area varies considerably year to year. The iNaturalist observations actually cover a wide portion of the range. So the dark gray you see is the total range of Joshua trees, and then any of the dots are locations that Joshua trees occur on that are observed on iNaturalist. So the blue dots are where we've seen fruiting happen, and then the red dots are where we've seen flowering but no fruiting. It doesn't mean that fruiting has never occurred there. It just means that iNaturalist might not have recorded it because no one ever came back to that site at any point. So I'm actually trying to supplement that data by also doing some field surveys. So I did a bunch of field surveys this spring and summer, where I just go out and walk along transect, see how many trees made flowers, and then I come back in like a month and a half and see how many of those made fruit. So 2024 was a pretty poor flowering year. There wasn't too many, but I did see 52 plots that had flowers on them, and 43 of these made fruit. And then once I collect all this data, I'll be using BART modeling to actually model what these moths are actually responding to. So BART modeling is a Bayesian additive regression trees. It's a machine learning technique used in many species distribution modeling. It's less prone to overfitting than other similar tools, and my modeling will be adapted from Yoder et al. Flowering model. So we take all this fruiting data, we combine it with all the weather predictors that we think are important for these species, and we put it in this BART model, and it'll spit out the best predictor list, and then these predictor effects that I showed like earlier with the flowering. So the factors that Yoder et al. Included was yearly rainfall, the minimum and maximum temperature, the vapor pressure deficit, and the year-over-year differences in these factors. And then I'm also going to include like quarterly and monthly variations in these predictors, sampling density, and data source. I'm working on that, but I haven't got it done quite yet, so I won't be showing that. But I have done some modeling just with these same predictors that the Yoder et al. Paper used. So there was 14,000 observations in that data set, and 748 flowering or fruiting records that were usable. So this is the predictors that it gave me. So the top 6 you see are the same from the Yoder et al. Paper. So these moths are responding to some of the same cues, except there was one additional predictor that the model said was important, which is the minimum temperature difference between the year fruiting was observed and the previous year. And then 2 of the predictors that it said were the same from the flower that the moths are responding to that also cue flowering, were the minimum temperature in the year fruiting was observed and the difference in maximum temperature between the year fruiting was observed in the previous year. So their curves are different, and I'm going to focus in on those. So on the left is the fruiting response. So it peaks below 0 there. So so it looks like moths are responding to temperatures when the year fruiting is observed, that moths are responding to temperatures below 0, while flowering happens when temperatures are above 0. So these 2 species that are in this mutualism are actually responding to 2 different things. Even though they're using the same cue, they're responding to 2 different parts of it, which warmer winters could disrupt this, and climate change could actually start to draw these species apart. Hopefully with more data, we'll be able to get a clearer picture on this. So these species are of conservation concern, and people in California and the West are very committed to helping preserve these species. So if we are going to help preserve the trees, we also have to know what's going on with these moths, and hopefully this will help illuminate some of those. So, just want to thank everybody in the lab that has helped me so far and everyone that has funded me. Does anybody have any questions? Yes.
Do you have any ideas for how you're going to monitor models besides necessarily just observations of the Joshua trees fruiting? Is there any idea of how you might get around that?
I have been trying to capture some moths and raise them in the lab, but has not been going well. I've not gotten any fruit. I need to capture the fruits like not too early, but not too late, and I missed it this year, so hopefully I'll be able to do it next spring, but we'll see. So. Okay. All right, cool. Thanks, guys.
To tell you today. So my talk is going to be about predation, climate change, and thermoregulation. And can everyone hear me well? Okay, good. So heat wave events are becoming more frequent and more severe. And so it's exposing many species to new levels of heat stress that are impacting their ability to persist into the future. Some of the ways that we can predict species vulnerability to climate change is by collecting data on that species' thermal traits and comparing that to the max temperatures in its habitat. And so we can calculate something like the warming tolerance. So this figure on the left side is showing us a thermal performance curve. And so the y-axis is telling us some kind of performance trait like feeding rate or heart rate, and the x-axis is showing us temperature. And there's a lot of useful information we can get from these curves, but one of the ones I'm going to talk a lot about is the upper thermal limit, which is where this line effectively terminates and the species can no longer survive at those temperatures. And so this is a really powerful method for predicting species vulnerability to climate change, but heat stress, like many other stressors, can have sublethal effects that can negatively impact organisms long before it reaches that lethal limit. And so for me, what I'm really interested in is understanding how these sublethal effects of sublethal heat temperatures can impact species interactions that can unveil hidden vulnerabilities that we may not have seen with the TPCs alone. And so in particular, what I'm really interested in understanding is how do heat wave events impact interactions between predators and prey, and how can heat wave events make prey more vulnerable to predation? And then more broadly, how does incorporating predator-prey interactions alter our predictions of species vulnerability? So to test these questions, I worked with Lotororia, which is a common marine snail genus that spends most of its time out of the water. And so it's exposed to a lot of the temperature variability that a lot of terrestrial ectotherms are exposed to. The two species I'm going to talk about today is Lotororia irrorata. It's a salt marsh periwinkle, and Lotororia angulifera, which is a mangrove periwinkle. So something really interesting about this genus and about subtropical and tropical intertidal snails generally is they're considered to have a really wide warming tolerance, a really big warming tolerance. So this is attributed to the fact that they're upper lethal limits. So here's the thermal performance curve again. A lot of these periwinkle snails can have an upper lethal limit that's over 10 degrees higher than their max habitat temperatures. And so they can have potentially a lot of buffer room between when temperatures should negatively impact them. And this is in really sharp contrast to what we know about a lot of tropical ectotherms that are actually predicted to be some of the most vulnerable species. So it's really interesting that these snails can potentially be, that appear to be not too vulnerable to warming. And so I'm first going to talk about our work in the salt marshes with irrorata. And so I'm going to tell you a little bit about how the predation and heat stress relationship is in a marsh. So heat stress and predator avoidance occur together. And so as it gets hotter during the day, the sun will come up, snails will experience higher heat stress, and they'll move down the grasses in order to thermoregulate. And so this is good. They can lower their body temperatures. But then when blue crabs, their main predator, enters the marsh, snails are perceived by both the airborne and waterborne cues from these crabs, and snails will actually migrate up the grasses to get away from them. And so in this system, there's a really clear trade-off between thermoregulation and predator avoidance. And so we had collected snails from the marsh, brought them back from the university, and created these mini salt marsh mesocosms. and we simulated heat wave events to test how snail microhabitat choice or height choice differed in between heat wave and non-heat wave treatments, as well as in the presence and absence of predators. And so we measured the snail body temperature and the height that it chose in these mesocosms. And so the first result I'm going to show you, so this is a density distribution of the snail heights that they had chosen. And the y-axis is telling us the heights of the snails, and the x-axis is telling us the density of the snails. Snails in blue are non-heat wave ambient conditions, and then snails in orange are those that have gone through the heat wave simulations for days. And so all this data was collected while these heat waves were happening. The first thing we see is, as you might expect, heat waves push snails to thermoregulate more, so they occupy a wider set of microhabitats to thermoregulate, and importantly, they move down a lot in order to cool their body temperatures, right? Now, the interesting thing is that when we look at our predator treatment, what we see is that in the presence of a blue crab, they actually completely eliminate this thermoregulatory behavior. And so the presence of a predator results in this restriction of microhabitat that ends up actually leading to the prey experiencing much higher heat stress than they would have experienced in the absence of a predator. And so we were getting ready to finish this experiment until an actual heat wave had passed through, and we were like, okay, great, not for me, but great for science. And so we decided to keep it running and actually try to capture this really intense heat wave that was passing through our area. And so the temperatures that the snails experienced in these buckets was quite a bit higher than what we were simulating, but it gave us a chance to test what would a really intense heat wave do to this interaction. And what we had found is actually kind of the opposite of the result I had just shown you. So actually in these higher intensity heat wave events, snails had all moved down the grasses regardless if a predator was there in order to thermoregulate. And so this is a part of a figure from our manuscript, but it's pretty much just showing us that the proportion of snails that became vulnerable to crab predation during heat waves reached near 100% during this high intensity heat wave event. Now, this is showing us that there can be this sort of vulnerability that as heat wave events are happening, prey can become closer to predators and increase their vulnerability to predation. At the same time, snails are also kind of making potentially a safer bet, right? Because experiencing your lethal temperatures, you have 100% chance of mortality, right? But predation is a probabilistic chance of dying. And so it's really interesting, and something I like to think a lot about are snails, they kind of have this physiological limit where if they're going to reach a thermal limit that might kill them, maybe betting your chances against predators might be a safer bet. And so I'm now actually going to switch to our work in mangrove systems. And the major differences in this system is that, and so in a lot of ways it's similar. Snails are exposed directly to air stress and solar radiation. But the big difference in this system is that the predation of these snails actually occurs entirely at night. And so this gave us a really cool chance to test what may be familiar for many people's systems is actually when predation and heat stress don't occur at the same time. And so we wanted to know, how does heat stress during the day impact predator vulnerability to predators at night? And so at night, crabs move into the mangrove roots, and then snails, and then at night, we actually figured out that these snails do a nightly migratory movement up the trees, several meters high. It's really cool. And so we collected snails from these systems. We brought them back to the lab and exposed them to different levels of heat wave intensities. And then we reset them out back into the mangroves and monitored their microhabitat use and the relative predation rates between snails that have gone through daily heat waves versus controlled snails. And so what I'm going to show you is a time-lapse video of what this looks like. So this is several hours condensed into like 20 seconds. I colored these snails with glow-in-the-dark paint because it's fun, but because to see it better. And so the snails in green are snails that have been just in ambient conditions, and snails in orange are those that have gone through a heat wave. We did a lot of treatments, but we actually just finished collecting this data about a month ago. And so I'm kind of just showing you the really preliminary stuff and what are the patterns showing us. So these snails in the orange are gone through the heat wave. The blue line is showing us the water line. And what we figured out is that snails that are within even a half a meter of the water can experience really high nocturnal rates of predation. So kind of keep that in mind while I play this. So something to notice that those that have been in ambient conditions are really active and they will move up the mangrove. While those that have been exposed to heat stress are kind of just stuck where they are, right? It looks like their behavior is really impaired from experiencing that heat stress. And so what we found with these experiments was actually that both the microhabitat choices that snails were making and their relative rates of predation were much higher for snails that have gone through a daily heat wave than those that have gone through that had just been in ambient conditions. And so the one thing I like to think about as I'm processing this data, maybe a great conversation we can have is, is this something like a choice? Are snails actually staying there because they've experienced so much daytime heat stress that they'll stay near the water and wait for high tide? Or is this actually some kind of like physiological inhibition? Like, are they actually physiologically damaged and they just can't really move up those routes anymore? And so now I want to briefly go into a bit of a broader picture about, I've been showing you so far the response of prey to predators, but of course it's a relationship between two species or several species. And so how does the predator's thermal tolerance relate to this? And so here's the thermal performance curve again now. So this is the mangrove periwinkle. It has an upper lethal limit of 54 degrees, and so it has about a 10-degree warming tolerance, a really big warming tolerance. These little graphs on the bottom are just showing us the habitat temperatures. Now, something really interesting that we found is that when we measured the thermal performance curve for the blue crabs, they also have a really high thermal tolerance relative to their habitat temperatures. And so they also have something over a 10-degree warming tolerance. And so this was pretty surprising and really interesting that it actually seems like both predator and prey have similar levels of physiological buffer room between current temperatures and their upper lethal limits. I'll just mention this briefly, again, as maybe a conversation point later, is that we actually measured also the thermal tolerance of another predator in the system, a pufferfish. And so I can't show you the graph because I wasn't able to get all the data for this species, but we found that its upper lethal limit is actually quite a bit lower. So the pufferfish has a pretty low thermal tolerance. And actually, it seems like within a 2 to 3 degree increase in the mangrove habitat, pufferfish would be pretty close to their lethal limit. And so I hope all this is to say is that the more predators we can incorporate in these kind of predictions, the more we see that the picture can be kind of, some predators have that persistence, some predators not so much. And I just want to briefly thank Chris Duane at the University of Massachusetts and Katie Eaton, who's giving a talk here as well, for helping me with a lot of these physiological questions and experimental designs. And so the major takeaways from my talk is that the fear of predators can increase the heat stress that prey experience. Heat events can also reduce the anti-predator behaviors that we've seen and increase the prey vulnerability to predators, both during the day when predation and heat stress are occurring together, as well as the carryover effects of heat stress during the day into the night. Some predators can have high warming tolerance, and so it seems that potentially blue crabs can be reaping the benefit of behaviorally impaired snails, while other predators seems like they may not be in those habitats for too much longer and may not be able to enjoy more snails. And more broadly, I just want to sort of talk about how incorporating species interactions with thermal trait data like thermal performance curves can really reshape our predictions of species vulnerability to extreme weather events. So I just want to say thank you so much for everyone who's helped me with these projects, to my advisor, Dr. Morgan Kelly, and the Kelly Lab family, and so many other people. Please see, our postdoc in our lab, Isabel Nalen, is giving a talk on actually the plasticity and transgenerational effects of heat stress on another intertidal copepod. And so please go to see her talk. And with all of this, just like to take any questions. If you can't read this, it's just saying, as climate scientists and ecologists, we're really excited when an extreme weather event happens during your experiment. We're not really excited when it keeps happening during all of your experiments. So it's, do we have controls anymore? But yeah, thank you. Oh, yeah.
So the experiment where you had the painted snails, which looked super fun, by the way, you had them, at least in the video, they were separated out, right? Could it be anything about the substrate that they were on maybe inhibiting their movement?
Yeah, so that's a good question. So we used mangrove roots that were both on the surface and structurally really similar for these experiments. So when we had set these out, we kind of had like a set criteria of how big those roots were and just like the relative diameters of them. The surface is pretty much the same between them, between these mangrove roots. But we ended up replicating this a bunch of times. And so that's what we're hoping to disentangle, is that if there was sort of any effect of microhabitat structure, we should hopefully tease it out by doing this across many roots. Yeah. I think I've seen you first.
Did you measure the mortality rate from predation versus heat stress during the different experiments to see how much their changes in behavior changed their mortality?
Yeah, like mortality from predators and mortality from heat stress.
Both, depending on what the
Yeah, we did. So in both of these experiments, the first one with the irorata, we did monitor, and we had maybe 1 or 2 snails that died just in those heat wave chambers. And this was a really cool thing because it kind of was telling us that they were really good at thermoregulating. So from heat stress, we pretty much had close to 0 snails dying from that. The mangrove experiment, because I was still experimenting with the treatment temperatures, there were some treatments where I actually had quite a few snails died. And in those treatments, I excluded those temperatures from our study because we wanted to just use temperatures that were sublethal. Yeah, I hope that answers your question. Yeah. And then I think, oh, sorry. Oh, yeah. No, thank you.
All right, our next speaker is Mariana Braga from the Swedish University of Agricultural Sciences.
Yes, thanks. Thank you for the organizers for having me. I'm feeling very fancy with the microphone because I have a PDF. Anyways, thanks everybody for being here. I'm Mariana, and I'm going to talk about this project that I'm working on right now. And I really hoped I would have results to show you, but methods development seems to always take longer than we think it will. So I don't have any results, but I still thought that it would be worth it to share with you the ideas behind this project. So my work revolves around evolution of species interactions, mainly host-parasite interactions, and butterfly caterpillars and their host plants are my favorite system. And yes, caterpillars are parasites. So a lot of my work is to compile interaction records from the literature for a given clade. So in this example, parrot butterflies, and put them together in a network like this. And then combining it with a phylogenetic tree, then we can try to reconstruct how that network evolved over millions of years, right? So a lot of my past work has been trying to figure this part out. But now let's say that we have observed this little butterfly caterpillar plant network, and we wonder not only how it looked like in the past, but also how it's going to look like in the future. So what kind of information do we need to make any sort of meaningful prediction? So we know that global changes in climate and land use are forcing species to move to find suitable habitats. And there's already a number of examples in butterflies where they have gone through geographical range shifts. And because species respond differently to these environmental changes, the butterflies won't necessarily find the same host plants in their new ranges. So interactions will be lost, most likely. But then what about formation of new interactions? And how can we go about trying to understand that? So the first kind of information that we need is where those species occur today. And luckily for me, Switzerland has a really good monitoring system, or 2 really good monitoring systems, one for butterflies and one for plants. And so I'm using their data, their occurrence data that is available in GBIF. And you can even get like higher resolution data if you contact them directly. And then I'm using joint species distribution modeling to infer the environmental niche for each species, for each butterfly and plant species. And with that, predict their future distributions based on different scenarios of climate and land use change. And based on that, then we can calculate what's the probability of co-occurrence between each pair of butterfly plant species. I'm saying species, but for the plants, we might need to go up to genus level because otherwise it gets too big. But it's still, it would be great if we could do species level. So, okay, so let's say that we have done this, and then we infer that this plant is going to disappear. It's not going to be available anymore for those caterpillars. What does that mean for those butterflies? Does it mean that they will go locally extinct because they don't have a host anymore? Well, now enters the second part. We need to talk about realized and fundamental host repertoires. So a host repertoire is the set of hosts that a species uses. And a butterfly plant interaction starts when a female butterfly chooses a plant to lay its eggs on it, and then that plant will be the host for its offspring. And so in this graph, in the x-axis, there are different host plant species, and then in the y-axis, there's oviposition preference. So so the female, which host plants they prefer. And the realized host repertoire is then the plants that the female lays eggs on and the offspring survives. So interactions that happen in nature. But we often don't observe is that the offspring, the caterpillars, can actually survive on a broader range of host plants. And this I call the fundamental host repertoire, analogous to realized and fundamental niche. And I call these plants potential hosts. So in our little example, the only information that we had, the only observed information we had, observed interaction was with this one host plant. But if we uncover the whole fundamental host repertoire, then we see that there's other two hosts. One, it's a hidden interaction. So they are already interacting, but we haven't observed it yet. So it's a false negative in our data set. And there's also a potential interaction with the host for which the caterpillar is already pre-adapted to. It is able to use that host, but the plant's not available, so they don't interact. Another way to visualize this is if we think about the overlap between the available plants and the fundamental host repertoire. So then the potential host is the host that, the caterpillar can use, but it's not available at the moment. But if it becomes available, then that interaction would be possible. So with that information, now we can update our prediction from local extinction to survival onto possible hosts. So I hope that with this little example, I convinced you that unraveling the whole fundamental host repertoire is really important to be able to make any meaningful predictions about future changes in networks. And then the question, of course, is how? How do we get at that? So you can, to get direct evidence, then you would have to bring caterpillars into the lab, rear them on different host plants, see if they survive. But that's a lot of work. So there's not that many studies that produce that kind of data. An alternative is to use phylogenetic inference to infer these potential interactions, and that's what we've been trying to do. So a few years ago, we published this model of host repertoire evolution. And so say we have a parasite, a butterfly tree, and then at the tips, the trait that it's evolving in the tree is a vector, and then each element in the vector represents the interaction with the host taxon. And then each one of these elements can take one of three states, 0, 1, or 2. And they represent no host, so it's not a host, potential host, and then actual host. And the way these states change over time, so host gain and host loss is a two-step process. First, the parasite has to gain the ability to use the host, and only after that it can actually start using the host in nature. And so based on a parasite tree, a host tree, which is optional, and then the host repertoires, the observed interactions at the tips here, so that's the data. Then what we do is we infer the ancestral state here, the ancestral host repertoire for the clade, and then the history of host losses and gains that have produced the observed interactions. So this is the paper where we proposed this model, and this is an example of an ancestral state reconstruction. So in the matrix, you have the observed interactions, and then over the butterfly tree, you have the ancestral states. But then what we're doing now is, oh yeah, and I forgot to say, this is implemented in RevBase. It's working. It has been used for other systems as well. There's R package. I haven't forgotten about it. There's an R package whose paper will come eventually. There's an R package also to make plots from this analysis in RevBase. But then now I'm implementing it in Tree People, which is a new software for phylogenetics. And the main thing now in this implementation for me is that it can take an input like this. So we have two interacting clades, and then in black we have observed interactions. So you know that that this guy interacts with that guy, for example. And then in the white cells, instead of saying that's no interaction, now I can say I don't know. And then when I do that, I can infer, at the same time I'm inferring the ancestral interactions, I can also infer the probability of these extant interactions. In other words, the probability that this host is in the fundamental host repertoire of that butterfly. So this is the overall vision. So combining the probability of co-occurrence that we get from joint species distribution modeling with the probability of the host being the fundamental host repertoire for a butterfly, and then combining all of these probabilities together to form probabilistic future networks for different scenarios of climate and land use change. So I'd like to thank all my collaborators. And then lastly, I just wanted to do some shameless promoting and say that next year I'm going to start as assistant professor at SLU, and I'll be looking for PhDs and postdocs starting in a few months. So come talk to me, and thanks. Have time, right? Yeah. Yeah. Yeah, so right now, the question is if we include the phylogenetic relationship between hosts in the model. And the answer is yes, you can incorporate any measurement of distance between hosts. I've only tested with using phylogeny. And then there's a parameter that you infer that tells you how much that that phylogenetic distance matters for gaining new hosts. So whether it's easier to colonize hosts that are closely related to each other or not.
I sometimes notice that introduced plant species will attract native butterfly species. And like there's maybe not such an obvious relationship between like native hosts and the leaves. So it would be really cool to see if like maybe phylogenetism doesn't matter at all.
Yeah. Yeah. Yeah, and I do think it maybe varies for different groups of butterflies as well. Yeah. I'm not completely sure what you mean, but my answer is I haven't looked at any traits, and I've only used species trees, and they have to be time calibrated. But yeah, we can talk later. Thanks again.
Okay, our next speaker is Digo Godinho from F Ciencias, Portugal.
Okay, thanks. Hi, thank you for coming. I'm going to talk about herbivore adaptation to metal accumulation in plants. And first I would like to show this picture. These are not soda pools. This is a river running next to an open-air mine in South Portugal. And even though this is a very harsh environment where contaminated by high amounts of metals, you can see plants growing here. And they can do this either by excluding the metals on their roots or by uptaking the metals, transporting the metals to their leaves and accumulating the metals there without suffering any toxicity. And even though this later strategy is much more costly metabolically for the plant, it also provides some benefits to the plants. And that's because some plants, termed hyperaccumulators, they can store in their leaves amounts of metals that are toxic to most organisms. And so this can be used as a defense against herbivores. And indeed, this elemental defense has been shown to have negative effects on herbivores, on many species, on laboratory and field studies. However, some herbivores may be able to adapt to these defenses and become less susceptible to them. Still, there are a few examples in the literature about this, and additionally, in metal contaminated environments, you can have both types of plants. So plants that don't have metals on their leaves and plants that accumulate metals on their leaves. And this creates an heterogeneous environment for herbivores. But how this heterogeneity affects herbivore adaptation to high amounts of metals in the leaves of plants, we don't know. Besides that, some herbivores avoid plants with high amounts of metals. And so they can escape this selection pressure. Nevertheless, other herbivores don't do this. And we don't know what is driving these differences. Maybe it's their recent evolutionary history. That's unknown. And so to try to answer to some of these questions, we used a system with tomato plants that can hyperaccumulate cadmium on their leaves, and the amount of cadmium that they accumulate correlates really well with the amount of cadmium provided in the soil. And herbivorous spider mites, Tetranicus urtica and Tetranicus evansi, that, as you can see in the right panel, they produce much lower number of offspring on plants exposed to cadmium than on control plants and exposed to cadmium. And in this talk, I will focus mostly on Tetranicus evansi, but if you want to know more about how these two species interact in the presence and in the absence of cadmium in the plants. And if you missed Inês' talk this morning, you can chat with her. She's really nice. And you can also go to Maria's talk Monday at 3 p.m. Okay, so the questions that we were trying to answer were if spider mites adapt to high concentrations of cadmium in the plants, if evolving in heterogeneous environments affects this adaptation, and if they are able to evolve avoidance of plants with cadmium in heterogeneous environments. And for that, we used experimental evolution, so from one ancestral population of Veteranix zevansi outbred population, we created 3 selection regimes, replicated 5 times each. And so one with plants without cadmium, one with plants with high amounts of cadmium, and one heterogeneous environment with the 2 types of plants. And we had the mites growing in this environment in discrete generations, and then after a while, we tested the performance on plants with and without cadmium and choice between those 2 types of plants. But this was after 2 generations of common garden in an environment without cadmium. So first, for the performance, we took mites from all the populations of the 3 selection regimes, and we tested the performance of individual females, mated females, in each of the environments, and we measured several life history traits. But in this presentation, I will focus on the number of adult offspring, as it comprises the effect of cadmium on the number of eggs laid, hatching rate, and juvenile survival. Okay, so let's first focus on the control selection regime, naive to cadmium. We can see that even after 12 and 33 generations, they have lower performance, so they produce fewer offspring on environments with cadmium, in orange, as we expected, but we knew about the ancestral populations. Now, if we focus on environment with cadmium, despite a slight increase in offspring production at generation 12, we can see that at generation 33, the populations growing in environments with cadmium, either homogeneous or heterogeneous, they produced the same number of adult offspring as the control populations. So was there no adaptation to the environment even after 33 generations? It could be. Or it could also be that the ancestral population lacked genetic variation for the response to cadmium in these traits, or that adaptation to cadmium involved different traits that we did not measure. For example, they could have evolved a slower metabolism that would allow them to better detoxify cadmium, but we wouldn't see that in our experimental design because this basically would increase their longevity and not their offspring production in just a few days. It could also involve some epigenetic regulation that was lost after the 2 generations of common garden. Then, if we focus on the environment without cadmium, we can see that the populations evolving in environments with cadmium, either homogeneous or heterogeneous, had lower offspring production in environments without cadmium as compared to the control populations. And this suggests costs. So we tend to think that they indeed were adapting to the environment, we just didn't measure the right traits. And also, this is in line with the hypothesis that they evolved a slower metabolism, because in the absence of cadmium, if they have a slower metabolism, they will eat less and get less energy and produce fewer offspring. But this is still an hypothesis to be tested. Then we also measured choice between the 2 environments. So we took the females from all the populations again, and we placed them in the middle of a plastic strip connecting leaf discs of the 2 of plants from the 2 environments, with or without cadmium. And then we followed the females for 48 hours. There were no differences in position between 24 and 48 hours, so we assume that they have chosen. But we saw no discrimination of environments with and without cadmium in the control populations, and also that this trait did not evolve in the populations evolving in heterogeneous environments. So they don't use this to escape plants without cadmium. This could be also due to a lack of genetic variation for choice in these populations. Or that they use other cues to discriminate plants with and without cadmium rather than tastes. So overall, cadmium seems to be a strong, cadmium accumulation seems to be a strong selective pressure for herbivores, at least for spider mites. And adaptation to cadmium in plants may involve changes in metabolism, but a slower metabolism also means slower host exploitation, so this is good for the plant still. But because spider mites were not able to rely on taste to discriminate between plants, they may still go to plants accumulating cadmium, so this herbivory is still a selective pressure for those plants to accumulate more cadmium. So, yeah, by now I hope that I convince you that metal accumulation in plants, it's a good driver of eco-evolutionary dynamics between plants and herbivores in metal-polluted environments. And yeah, thank you. I would like to thank my former team, the Mite Squad, and the organizers of the symposium, and you for listening.
Great talk. So, do you know what other traits of the plants might be changing with high doses of cadmium? Are there any changes in like nitrogen availability or maybe other like chemical defects like nicotine that they might be producing?
Yeah. So, great question. Thanks. So we tested a lot of traits in the plants previously, and we didn't see a lot of changes. We did observe shifts in sugar content, but then we tested this. So in artificial diets, different concentrations of sugars and different concentrations of cadmium, and we saw that the effects, it's driven by cadmium and not by changes in the plant.
So you're hypothesizing that there might still be a kind of effect on the metabolism of having cadmium in plants, right? So if we assume that's true, and the spider mites can't differentiate or don't differentiate between cadmium plants and non-cadmium plants, does that mean that if you are a plant in an environment with cadmium in it, if there are enough kind of hyperaccumulators around, you might kind of cheat the system and decide not to accumulate because the other plants are kind of depressing your spider mite population?
Yeah, that could be, but hyperaccumulation is a rare trait. Even though it spreads through the plant kingdom, it's not the most common strategy. I think it's very, very costly for the plant to do all of this. It involves many different transporters and accumulating the metals in the vacuoles without suffering toxicity. So it requires a lot of energy.
I guess a follow-up to that then, how much do the spider mites really move around? Like, how much can they travel?
Well, they can disperse between plants by walking. They can also disperse through wind. So yeah, they can spread around. Yes. Thank you.